In this video, we will discuss about how one can use the biological advancement to improve the CAR-T efficiencies. As per the current trend, that is till 2025, most of the second generation CAR-Ts are been are in a clinical trial stage. So, however, with the advancement in the biological knowledge such as molecular biology, synthetic biology, and gene transfer technologies uh, improvement, one can further improve the CAR-T efficiencies against the tumors, or one can also increase the survival durations of these CAR-Ts within the host. So here some of the examples are shown in this figure, that some of the things can be overexpressed and some of the things can be knocked down. So we will first discuss about the knockdown. So in terms of the knockdown, they talked about the DAG kinase, that is DGK alpha and DGK zeta. So that we have discussed when we are understanding, when we are discussing one of the modifications in the CAR-T in our previous paper discussions, that knocking down of DGK kinase will improve the CAR-T cells survival efficiencies, survivals rate within the mice, and at the same time it will also increase its anti-tumor efficacies. So what it does is that with the DGK kinase phosphorylations, it will affect the T cell signalings, and sometimes this DGK kinase will also cause over activations of the T cells, and that might leads to the other problems, such as the immune system is uh, causing some of the side effects. So by knocking down this DGK or DAG or DAG kinase, one can improve the CAR-T cells survival times and its anti-tumor efficacies. And what this paper is suggesting, or this review paper is suggesting, is to knock down these TETs that are found in the nucleus, that is finally responsible for the overactivations of the T cells. At the same time, it will affect the T cell signalings. And one of the conditions that CAR-T cells might encounters while dealing with the solid tumors is the environment that is deprived of the nutrients. So in order to take care of that, what this one of the approach that can be used is that overexpressions of the this nutrients transporter, such as the glucose transporter overexpressions and this amino acid transporter overexpressions, and this will help these CAR-T cells to survive even in the nutrient deprived conditions. And what it will does is it will lead to the upregulation of this FOXP3 that will cause the epigenetic patterns changes and results in the enhanced survival of the CAR-T cells even in the nutrient deprived conditions. And the other things that is shown here is about this while increasing these nutrient transporters, it might be possible that there is some side product that has formed, and that is detrimental to the CAR-T cells health, such as the acetyl CoA might forms this lipid droplet or cholesterol esters that is not good for the health of these CAR-T cells. So these things, ACT1, which converts this acetyl CoA to lipid droplets or cholesterol esters, can also be knocked down to further enhance the results. And in terms of the overexpressions, this in order to enhance its anti-tumor efficacies, this co-stimulatory domain 4-1BB can be enhanced. Its activations can be enhanced using the overexpression of this PGC1 alpha, and this will cause the overexpressions of the overactivations of the, and this will cause the activations of the T cells that might target the tumors. So some of the things that can be overexpressed in these pathways is this PGC1 alpha, GOT2, and in terms of this T cell death, in order to avoid this T cells death, this OTC and this ASS, these things needs to be overexpressed. And in terms of the things that needs to be down regulated in terms of the receptors is this AE2 receptors. As it will does is it will help in the movement of this it will help in the transport of this lactate into the cellular matrix, and that needs to be prevented for the internal functioning of the CAR T cells. So these are some of the changes that can be done to increase the CAR T cells efficiencies, not just in terms of the survival rate but also in terms of the targeting to the also in terms of its targeting efficiencies against the efficacies against the tumor. So that's all for this video. If you need to understand more about the topic in details, you can go through the reference that has been provided in the description. Thank you for your time.